It's my great pleasure to join the seminar and present some of our recent work on incorporating the functional genomic annotation to improve prediction, fine mapping, and mapping cell types relevant to the traits. Okay, all right. So I guess my journey of exploring the utility of functional annotation starts with investigation of the genetic architecture of complex traits using the GWAS data. As all you know, the GWAS has been quite successful in identifying the genetic variants associated with traits. Perhaps one of the most important messages that we learn from GWAS is that the complex traits and common diseases are very polygenic. For example, as shown in this results from the latest schizophrenia GWAS study, they identified nearly 300 risk loci at the genome-wide significance level, and they predicted that thousands of more loci will be identified when the sample size further increase. And the reason this prediction is made is based on the observed genetic architecture of the traits. Using a Bayesian methods that I talked about in a moment, we are able to estimate what proportion of the genetic variance is explained by the small, medium, or large effect. And schizophrenia is here. As you can see, most of the variants are explained by small effect. And of course, the genetic architecture are not the same between any two traits. Type 1 diabetes has some major gene effect in chromosome 6. But overall, you can see that the complex traits are very polygenic, and their genetic architecture is dominated by many, many small effect variants. So that leads to a question: why these common diseases, complex traits are so polygenic? And maybe the answer lies in the evolutionary genetics. If we consider the neutral mutation due to the random drifts and the mutation, the allele frequency just fluctuating along generations. But if the mutation is deleterious to the fitness, under natural selection, the allele frequency tend to decrease over generations of selection. And if we look at the snapshot of the last generation, the deleterious mutations tend to have a lower frequency than the mutual mutations. So that leads to a signature of natural selection if the trait-associated variants are likely to be deleterious mutations. The mutations with larger effect on the trait are likely to be more deleterious. Therefore, under natural selection, we expect them to be kept at a low frequency. And in the common allele frequency spectrum, and that's the spectrum we identify from GWAS, the effect sizes tend to be small. Indeed, we observed that kind of the relationship or pattern in common diseases, like in schizophrenia, the large effect observed in the very small minor allele frequency spectrum. However, by experimental design of GWAS, the power for detecting the rare variants are quite limited. So only the large effect can be detected. That presents a challenging of ascertainment bias. So in our first study, we trying to model this relationship across all variants in the genome without doing any selection using this mixture model, where the effect size or the variance of the effect is modeled to be a function of the allele frequency, to PQ, to the power of S. So if the S is negative, that represents this negative relationship between the effect size and minor allele frequency. And in this study, we found that if we apply these methods to complex traits, most of the complex trait we analyzed has a negative S value. So that indicates the prevalent action of natural selection. And then in the subsequent study, we go ahead and look at this signature of natural selection in different functional categories, like the transcription start site, conserved region, coding region, enhancer promoter, and regulatory regions. And what we found is that both the habitability enrichment and the signature of selection seems to be varying across functional annotations. And that suggests the effect size distribution are likely to be different between functional annotations. We think that's an important message or lesson that we learn from studying the genetic architecture. And then the following question is, what can we do with this knowledge? So this is what my talk today really focused on, is how we use this knowledge that the effect size distribution are likely to be different between annotations to improve the polygenic prediction, improve the power of fine mapping, and use the functional annotation to identify the cell types relevant to the complex traits. So for each of the topic I talk about the methodology we used and what we have found in the data analysis. Okay, first to talk about the polygenic risk score prediction. And the polygenic risk score, or PRS, predict the genetic liability of an individual to disease, or PGS is the application of the risk score alleles. So this PRS is a linear combination of the genotypes and weighted by the SNP effect. So the key questions in this equation are which SNPs should be included and what are their weights. The conventional methods called C plus PT or clumping p-value thresholding only select the subsets of SNPs that pass the genome-wide significance threshold and only use those SNPs with their GWAS effect for prediction. The newer methods tend to model the LD between SNPs and fit all of the SNPs simultaneously in the model. And different methods, many different in using different prior distribution to account for different genetic architecture. Like if you use the normal distribution for the SNP effect, that's what LD-prat-in for S-block do. And other methods use the mixture distribution of spike and SPLAB, or even a multiple component mixture distribution. To our experience, the best methods for PRS prediction is the method that best describes the distribution of SNP effect that is flexible to accommodate different genetic architecture of the traits. As we have seen these observe the genetic architecture for different traits, and they have different architectures, and that's the main reason that we like to use this multi-mixture distribution to account for these complex architecture. In the initial study, we use this S-based R method and work on the summary statistic model to unleash the power of using the summary data. And very quickly, the summary data model can be derived from the individual level data model. If we start with this classic model, the phenotype is the consequence of the genotype matrix times the SNP effect plus residual. If we multiply both sides by 1 over n, genotype matrix transpose, where the genotype matrix is standardized, then we get this equation. And you might recognize that the left-hand side of this equation is the GWAS marginal SNP effect B, and the middle part, 1 over n x prime x, is the LD correlation matrix that you can calculate from an independent reference sample. And even though in the original model, the residuals are uncorrelated, Here, the summary data model, the residues has a correlation structure that is proportional to the LD correlation matrix. And in S base R, we assume this joint effect has a mixture of multi-components. And in our recent work, we extended this method by incorporating the functional annotations. And we incorporate the functional annotations through this hierarchical prior. So this is the prior used in S-base R, where each SNP has a mixture distribution, but all of them has the same mixture distribution regardless of where the SNP is. Now, we're trying to incorporate the SNP annotation into this mixing probability parameter by assuming this function of this mixing probability is the linear combination of SNP annotations. And we use this probit link because this probability has the 0, 1 scale, and this annotation effect cover the whole real line. And essentially what it says is the decision that we make to put the SNP into different distribution depends on the annotation of the SNP. If the SNP has multiple important annotation, for example, if the SNP is in the coding region that is conserved across species, it's likely to have a large effect. And we use this generalized linear model because it's convenient and it naturally accounts for the overlap between annotations. And we know in the human genome, it's likely to have lots of overlap between annotations. And it also naturally accounts for both binary and quantitative annotations, like if the SNP is in the coding region or not, or the LD score or allele frequency of the SNPs can also be used as annotations. Another trick we did in S-base RC method is to use the low rank model, and that would allow us to computationally efficiently fit many SNPs. Like we tried 7 million SNPs or even more to fit them in the model. It works as follows. So first we find approximately independent LD blocks across the genome. Based on prior study, there are about 1,500 LD block in the human genome of European ancestry. And in each LD block, we fit this summary data-based model. And now we have the LD correlation matrix between the SNPs in the block. First, we do eigen decompositions to factorize the matrix into the matrix of eigenvectors and the diagonal matrix of eigenvalues and the transpose of the eigenvectors. Now, the trick is that we don't use all of the eigenvectors or eigenvalues. We only use the eigenvectors that corresponds to big eigenvalues. And then if we multiply both sides by this eigenvalue and eigenvectors matrices, and we can end up with the new equation where the dimension of the y variable can be substantially reduced, but we reserve the dimension in the SNP effect. Essentially, what this model said is that we are using we only use the principal components of the LD matrix, and by doing that, it collapses the mutually dependent marginal effects into a subsets that are independent of each other. So in the residual, we have this diagonal structure for the residual variance. And you might wonder how much pieces we need to keep to explain most of the variance. It turns out we can get rid of the 80% of the eigenvectors and only use the top 20%, and that could be enough to explain 99.5% of the variance in LD in the European ancestry. So by doing that, it becomes more efficient and also improve the robustness, because now we can estimate or sample the residual variance from the MCMC. So these are just the simulation results to demonstrate the improved model robustness. These are the two key parameters we interested in the model, SNP-based habitability and the homogeneity. So that's basically the proportion of SNPs with non-zero effects. And we are making this simulation to challenge the model by choosing a proportion of the SNPs that are not overlapped in the meta-analysis. So that would create the problem because the residual variance covariance structure will be distorted if the proportion of overlap is not 100% in the meta-analysis. And as you can see, when they drop down to 0%, these parameters estimation is biased. But however, compared to the other methods like LDPRA2, we can keep the estimation precision of these parameters better. And that's owing to this low rank model and modeling the residual variance. So as you can see, the residual variance will be inflated if the overlap is low. And the residual variance is just a nuisance parameter in our models that we don't care much about its value. So in another word, we use this residual variance to absorb any data quality issue. All right, how does it perform? Of course, we do simulation and we did both within ancestry and cross ancestry prediction. Here, due to the time, I just show you a results for our cross ancestry prediction. So the goal is to use the GWAS data from the European ancestry and predict the East Asian ancestry in UK Biobank. We compare our methods to a method called the PIRCSX. This method doesn't incorporate function annotation. Instead, it used another data set of the target ancestry to enhance power. In our case, we used Biobank Japan, which has the same ancestry with the East Asian sample in UK Biobank. And using these methods, we observed the improvement of prediction accuracy by 17% across different traits. Now, the question we want to ask is, how does the incorporating functional annotation compare to using additional GWAS datasets? And here is the result. We looked at with or without annotation at the low density SNPs or the very high density SNPs. The key result is that when we use the high density SNPs with annotations, using the European data alone improved the prediction accuracy by 16%, already comparable to the 17% with additional Biobank Japan data. Now, if we use both Biobank Japan and the function annotations, we achieved the improvement of 33% across traits, almost double the prediction accuracy observed in the PIRCSX method. So I think that results demonstrate the benefits of incorporating annotations that it's comparable to increase the sample size at the GWAS level. Another interesting finding we found is the interaction between SNP density and the annotation information. So the result shown in this plot is the improvement of the prediction accuracy when using annotations at 1 million HiMAP3 SNPs, and the y-axis shows the same thing, but at the 7 million imputed SNPs. So if the improvement of annotation is independent of the SNP density, we should observe the results on the diagonal line. However, for most of the traits, if not all, that we analyzed, the improvement is appear to be higher when we use the high density SNPs. And why is that? I think the explanation is that when the causal variance is not observed, we can use the SNP to tag the causal effect. But the SNP in LD with the causal variant may not share the same annotation, so there might be a mismatch between the annotation and the causal effect. So that suggests it had better to use the very high density SNPs when we incorporate the annotations, and also further suggesting the potential benefits of using whole genome sequencing variants with annotations. In summary, in terms of methodology, we developed the methods using multi-component mixture distribution and incorporated the functional annotations to allow the annotation affecting the probability of the SNPs belonging to each of the component distribution. We found that the annotation data provide a comparable benefit to additional GWAS datasets for improving prediction. And the significant interaction between SNP density and annotation information suggests the potential benefits of using whole genome sequencing data with annotations. The second part is about incorporating annotation to improve file mapping. So there is a lot of file mapping methods available. FileMap and SUSY are the two predominating methods. However, most of these current file mapping methods focused on the GWAS region only or GWAS loci only. Typically, they identify the GWAS loci based on the significant threshold and open up a small window like 1 or 2 megabase and only do file mapping within the GWAS loci. We think the problem of doing that is we ignore the rest of the genome that potentially also includes the causal signal. And also, if you restrict the file mapping within the GWAS loci, the prior parameters in the hierarchical model may not be estimated very well. And it's hard to incorporate the function annotations because the function annotations does not only exist within the GWAS loci, but also across the whole genome. So we think it might be best to do the file mapping across the whole genome, along with the genome-wide function annotations. So this is the motivation of the project, and basically we use the GWAS summary statistics across the whole genome with the genome-wide LD blocks and incorporating the genome-wide function annotations. We repurposed the SBaseRC used for prediction. Now we use it for file mapping, and basically it's the same machinery, and we can calculate the posterior inclusion probability as the probability that the SNP come from the non-zero component. And we can construct the local credible set. We can also construct a global credible set that is defined as the set of SNPs contain a certain proportion of the causal variants. And we are able to do that because we are estimating the number of causal variants and the genetic architecture at the same time when we do the file mapping. And it's also because we're estimating the genetic architecture, we will also be able to predict the power for future study given a sample size. And I think that's a cool thing to do. Such a power calculation exists in GWAS, but does not exist for file mapping yet. Okay, the first results is using simulation to demonstrate the benefit of doing the genome-wide file mapping versus the file mapping restricted within the GWAS loci. So here we do a simulation under the standard sparse genetic architecture. So we random sample 10,000 causal SNPs as causal variants. And we compare the genome-wide file mapping with the GWAS loci-based file mapping. It shows the power at different PEEP thresholds. On average, about 30% of the causal variants we found are actually outside of the GWAS loci. So if we only restrict it to GWAS loci, you were likely to miss out 30% of the causal variants, potentially can be identified. What about in the real traits? So we applied these genome-wide file mapping methods to 48 independent traits in the UK Biobank. Again, similar to what we found in simulation, about 70% of the variants we found are within GWAS loci, and there are 30% outside the GWAS loci. Well, you might think those variants outside the GWAS loci are tend to have small effect, so they may not be important. So we estimated what proportion of heritability do these variants explain. Indeed, these variants tend to explain a smaller proportion of heritability, but still, those 30% outside of the GWAS loci putative causal variants together can explain 15% of the heritability captured by file mapping. So it indicates they tend to have small effect, but they are not trivial. And then we look at how much the functional annotation can help with genome-wide file mapping. So first, we look at the calibration of PEEP, because PEEP is the key statistic for file mapping, and we want to see how well the PEEP can capture the true proportion of the causal variance among all positive results. So here we did the standard simulation, but just to show you the results when we further challenging the methods by simulating some difficult scenario where the causal variants are sampled only from a high LD and high MAF malleable frequency region. So that would create a loss of LD between the causal variants and the SNP markers, and potentially LD between the causal variants as well. And in this challenging scenario, you can see that the PEEP are not well calibrated in the other file mapping methods. So the PEEP does not correspond to the correct values for the true discovery rate. So those are the fraction of the causal fraction of the variance above within this PEEP being a truly causal variance. The perfect calibration will be on the diagonal line. And these existing methods tend to have an inflation. They say the PEEP is 0.9, but the true discovery rate is much lower than 0.9. And when we use S-base RC and incorporating these LD and MAF functional annotations into the model, we get a very good, a much better concordance. Of course, we don't tell the model which LD and MAF categories contains the causal variance. We let the model decide the weights of each LD and MAF categories by itself from the data. And then, so the people basically assess the false discovery rate, how well the model managed the false positives. We further look at of the file mapping between different methods. Here, the power is defined by the proportion of the causal variance can be identified from the credible set or single PEEP SNP from different methods. And as you can see, depending on the PEEP cutoff, the S-base RC genome-wide file mapping has a clearly higher power compared to the local file mapping method. And we further look at the resolution, so that quantified by the credible set size. What we are saying is we not only want to identify more causal variants, but we want to identify those causal variants with a smaller credible set size. Indeed, the genome-wide file mapping tend to have a smaller credible set than compared to the other methods. We also look at the precision. So for the variance that does not pinpoint the causal variance, we want to see how far away it located to the actual causal variance. So this is measured by this precision, the distance between the identified variance and the nearest causal variance. And we found that the ping generates a much smaller distance compared to the other methods. Interestingly, this polyfount plus SUSI is the file mapping method also incorporates the function annotations, but they did in a stepwise manner. So they first use LD score regression to estimate the annotation heritability enrichment, and then incorporate that into the SUSI file mapping method using the SNP-specific prior. But it does not do as good as the genome-wide file mapping. And we think the reason might be doing the unified model in a single-step analysis might be better than the stepwise analysis. Okay, so that's simulation. In real data, we can't really look at the power because we don't know where are the causal variants. What we looked at is the enrichment of the functional annotations in the file map, the variants compared to the genome-wide significant SNPs and all SNPs. So this is the results compared to the GWAS clumped SNPs or all SNPs. The file map, the variants are more enriched in the functional important annotations and depleted in the repressed regions. And we apply these methods to the schizophrenia latest GWAS dataset, and we identified about 13 SNPs with high PEEP values. 5 of them are novel, and 3 of them are missense variants. So next, I'm just trying to show 2 examples in schizophrenia file mapping. One is this famous gene SLC 3988. This is one of the first genes that identified in the earlier schizophrenia GWAS study. And now when we apply these file mapping methods, it pinpoints the likely causal variants. And this has been well studied in the prior functional studies. And interestingly, you can see that this likely causal variance has the multiple important annotations, such as conservation across primates and mammals, and it's also a non-synonymous variance. And we also identified a secondary signal that these variants also tend to have important annotation as the conserved variants. Another example is a novel discovery. This ACTR1B gene is not reported in the schizophrenia Nature paper because from the GWAS, the top SNP barely passed the genome-wide significance threshold. And if you run the SUSI-INF, so that's the SUSI plus the infinitesimal component, this SNP is not does not have a very high PEEP. But in our genome-wide file mapping with functional annotations, this SNP has a high PEEP value. And we further look at the association of the gene to the schizophrenia, and we found this highlighted in the proteinomic association study and also protein QTL localization analysis. And this variance is a single cell eQTL for the gene in excitatory neurons. And that's also a cell type implicated in the cell type mapping analysis for schizophrenia. And this variance also has important functional annotations, and it's the missense variance for the gene. So putting all this evidence together, it seems to suggest that this is likely a causal variance for the schizophrenia. Okay, another thing I think it's cool to do is to predict the power for the future study given a sample size. The question we ask is, what sample size do I need to identify a certain proportion of the causal variance, or the causal variance can capture a certain proportion of the SNP-based heritability? Put the long story short, how we do that is we can derive the sampling distribution of PEEP given a sample size and given the estimated genetic architecture. It turns out the PEEP can be written as this expression, and in this expression, it has this constant that depends on the genetic architecture, and the only random variable is this chi-square variable depending on the sample size. So if the sample size is small, you can see the sampling distribution of the PEEP has a higher density in the low value. And when we increase the sample size, the distribution of people are moving towards the higher values. And we apply this to a number of real traits, including the simulated traits. So from the genetic architecture estimates, you can see that they can have different SNP-based heritability for different traits: height, Crohn's disease, schizophrenia, high-density lipoprotein. And they also have a different polygenicity, like the proportion of SNPs with different effect size looks different across different traits. Using those information in our prediction formula, we are able to predict the power to detect all causal variants, or the power to detect the proportion of the SNP-based heritability captured by the fine mapping credible sets. And so the x-axis shows the sample size in future GWAS, and y shows the power. The line shows our predicted value, and the dots represents the outcome from two GWAS study with different sample size. And overall, looks like the predicted value is close to the observed value. And interestingly, as you may see, that when the GWAS sample size is very large, so this would be 10 million, we still can't identify all of the causal variants for most of the traits. And this is because many traits has the very small effect, and probably even though we sequence everybody in the world, we still can't identify all the causal variants. However, when the sample size becomes quite large, like 5 million, we will be able to fine map the causal variants that capture most of the heritability. For example, for height, so that's the brown line. In 2022, Lokayengo presented a study that the G was for height using 5 million individuals, and he found almost all of the SNP-based heritability can be captured by the GWAS hit, and that's kind of consistent with what our prediction from the fine mapping analysis. This table summarized our prediction for the schizophrenia when the sample size further increased. So this is where we are in the first line with the 54,000 cases and 77,000 controls. We can identify more than 200 credible sets, and they altogether explained only 4% of the heritability. But when we further increase the sample size to 550,000 cases with equal number of controls, we expect to identify 10% of the causal variants. They can explain 50% of the heritability. And when in the future, if we are lucky enough to have 1.4 million cases and controls, we will be able to fine map 80% of the heritability. So in summary, we used SBSRC for fine mapping, incorporating functional annotations, and it appears to help with managing the false discovery rate, increasing the power, reducing the credible sets, and one key point we're trying to make is that it might be the time to move the fine mapping from GWAS loci only towards the genome-wide scale, because we can uncover the extra 30% of the putative causal variants, and they might account for 15% of the heritability on average across traits. And doing the genome-wide fine mapping also enables calculation of power for future studies, and that's might be useful for the experimental design of future fine mapping studies. Okay, in the last, I like to talk about these function annotations if you generate from the single cell study, and that can be incorporated with the GWAS data to fine map the cell types or developmental stage relevant to the trait. So the two commonly used types of single cell sequencing data are single cell RNA-seq and the single cell ATAC-seq. So the single cell RNA-seq tells us which genes are highly expressed in particular cells, And the ATAC-seq tells us whether the genomic region are accessible, whether it's open in a particular cell type and even developmental stage. So combining the GWAS with the single cell sequencing data provides insights into where and when these disease genes and variants act to influence the disease risk. And these two types of data can be collected independently. And the single cell data can even collect it from the healthy cohorts or different species like the mouse. This is just an example highlighted in the that schizophrenia Nature paper. They look at the cell type from human data and the mouse data, and uh, and they integrate with the GWAS and try to see what cell type are implicated from this integrative analysis. And interestingly, first, when the GWAS sample size increase, you can see that it's the similar cell type that consistently been discovered with more significant signals. And secondly, the cell type prioritized between human and mouse data are kind of similar. So these cell types, although they don't have exactly the same name, they are ranked based on is the excitatory neuron, inhibitory neurons, and then clear cell types. Yeah. So it suggests that we can observe the consistent results with increasing GWAS power, and we will be able to even leverage data from model animals such as mouse to provide insights into the cell types relevant to human traits. There are many methods available to do this integrative analysis, and this is the fast-moving area. Probably you see new methods coming out every week. However, we feel like there is a lack of a comprehensive comparison between methods and the evaluation on what factors affecting their performance. So this is where our study starts. We're trying to first summarize the common strategies, integrate the GWAS with the single cell gene expression data. Although there are many methods, but we think broadly they can fit in these two main strategy. One is we called single cell to GWAS strategy. Another one is the other way around from GWAS to single cell strategy. I talk them briefly one by one, and then we compare different methods and look at what factors are affecting their performance. So this study has been carried out mainly by my students Ang Lee, and now is a postdoc in the Oxford University. So first, the strategy is a single cell to GWAS. It starts from the single cell expression count matrix. So this would be the number of cells by number of gene matrix. Each entry is the gene expression level in a cell. And because there are many cells, a common way to do is to aggregate the counts of cells within the same cell type if they are pre-labeled based on the marker genes into this cell type by gene matrix. And in each of the entry now is the total expression of the gene across the cells within the same cell type. And people applied cell type specificity metric to come up with the genes that are specifically highly expressed in a particular cell type. And there are many metrics available. A common one is EP, expression proportion. Basically, just to calculate the mean expression of the gene in a particular cell type relative to the sum of the mean expression across all cell types. And then you can get the distribution of this EP score for across genes for a cell type. And people often take the top 10% genes as the cell type specific gene set. And this method is also called the TDAP, the top 10% expression proportion. There are some other methods available as well. ES-meal is the method trying to calculate the mean across all these statistics. CPO is a method not only look at the high mean expression, but it also look at the variance of the expression. So it's looking at the genes not only highly expressed in the cell type, but also stably expressed with a small variance. Anyway, usually people take the 10%, top 10% of the protein coding genes. So that would be 2000 genes as a cell type specific gene set. And then we are ready to integrate with the GWAS association signals to implicate the cell type relevant to the trait. And there are two main type of methods to integrate with the GWAS association signals. One is at the gene level, or the magma gene set enrichment. It's looking at whether the cell type specific gene sets are tend to enrich in the GWAS gene-based association signal. Another type of method is looking at the LD score regression, heritability enrichment, to test whether the heritability is enriched in the cell type specific gene set compared to the rest of the genome. So that's start from single cell towards the GWAS. Another strategy is start from the GWAS and moving towards the single cell for integration. So from the GWAS, we can do the gene-based test to prioritize genes associated with the trait or disease. Commonly used gene-based tests include magma and MBAC combo. So these are our own methods that account for the masking effect. And after we have the gene-based test, we can then we can come up with the trait associated gene list. Usually it's 1,000 genes. And then people look at how does these total expression of these 1,000 disease genes in the single cell data, and they compare to the control genes total expression in the same data set, and they call this as the control score, the disease scores of the cell versus the control score of the cell. And if the disease score is significantly different from the control score for the cells within that cell type, then they declare this cell type is associated with the disease. And this method is known as the single cell disease relevance score. So first, we want to compare these methods. And of course, it's very hard to do in the real data because we don't know which cell type are truly causal to the trait. So we first set up these ground truths for the method comparison. We selected 38 complex traits and disease with high GWAS power, and also look at the 33 cell types across different organs in whole human body. And first, we select the true positives by looking at the publication count, where the cell type is well studied for the corresponding traits, and we selected these as the true positives. And then we select another cell type that is least relevant to the trait as the true negatives. And then we compare different strategies like GWAS to single cell strategy and single cell to GWAS strategy, compare their power and false positive rates. These two strategies on average do not seem to differ too much in terms of power. And we found if it might be best to combine both of them using this Cauchy combination approach. So this is kind of the a novel approach initially people used to detect the rare variants. The mathematics, it's a bit complicated, but it turns out the application is simple. You just plug in the p-values from each test statistic, and the Cauchy combination methods will produce the single p-value that already account for the correlation between the test statistics. And when we use this Cauchy combination, we found the power is maximized compared to each of the strategy. And the nice thing about that is does not introduce any inflation in the false positive rate. Then we look at what factors affecting their performance. First, these single-cell to GWAS strategy requires to calculate the cell type specificity score, and there are different metrics available, like the EP, differential expression test, or SIPO. So first, we look at which specificity metric can best identify the marker genes for the cell type. So this shows the distribution of the ranking of the marker gene across different cell types. And apparently, this simple methods, differential expression test, t-test, can best identify the marker genes for the cell types. However, when we look at the power to identify the cell types for complex traits, the best methods can identify the marker genes for the cell types. It's not necessarily the best methods to improve the power. It looks like this SIPO, regardless using the algebra regression or the magma gene set enrichment analysis, it tend to have a higher power than the others. And second, we look at the origin of the species for the single-cell RNA-seq data set, whether it comes from human or mouse. And in our data set, we use this tabula sapiens or tabula muris from the same group. Although there are more cells from humans, it, it shows that the power doesn't differ significant difference between the two data sets, suggesting using the mouse data is a promising approach to do the initial analysis to study the human cell type trait relevance. And then we look at the cell type diversity impact on the power. Here we looked at the atlas data where include many cell types across different organs and compared to the power using a single organ data sets like liver, lung, heart, and we looked at the different mapping strategy. And the x-axis shows the cell type trait pairs. And the results shows that in most of the case, the atlas data give a higher power than the single organ data set. That just suggests that the cell type diversity really matters. And we think it makes sense if you think about from either of the strategy, we need to calculate the cell type specificity score. So that depends on what are the background cell types in the data set. Or we need to compare the disease score with the control scores for the cells within the cell type. And that also depends on what are the background cell types available in the data sets. So if all of the cell types are likely to be causal to the trait, there is not much contrast between the cell types, and that will reduce the power. Okay, and our last bit of results is about um, whether the GWAS sample size or the number of cells are more important to this integrative analysis. And we looked at the three traits: height, type 2 diabetes, and coronary artery disease, because there are a different sample size GWAS data available in the public domain. So first, if you look at the sample size of GWAS, it looks like still have a great impact on the power. When we increase the sample size, you see almost a linear increase in power. But when we look at the number of cells within the cell type, except for the coronary artery disease, it's a big impact. For the other traits, it looks like even though the number of cell type is small, number of cells within a cell type is small, like as small as 20 or 50 cells, we will still be able to identify the significant cell type for the trait. So it suggests that at the current stage, it seems the GWAS sample size is more important than the number of cells. So these are the key observations from our benchmarking analysis. The effective trait cell type mapping requires more than just identification of the cell type marker genes. And using mouse and human data produce consistent results. to improve power using a data set with diverse cell types or using a GWAS with high sample size is seems to be more important than using more cells. And to maximize the power, it's best to combine the two strategy. And the Cauchy p-value combination provides the convenience way to do that. Um, this is my last two slides. I just want to showcase one of the results when we apply these methods and to gain the biological insights into psychiatric disorders to ask a question, which cell type and in what developmental stage the disease genes and variants act to influence the disease risk. And incorporating these cell type and developmental stage annotation, whether they can improve prediction accuracy. So we combine this schizophrenia GWAS study with this single cell study, where they have collected the single cell ATAC-seq and RNA-seq from individuals with different developmental stages, all the way from fetals to childhood, adolescence, and adults. We used this SBSRC framework, but now we incorporate baseline annotation plus the single cell and stage developmental annotations. So the gist is that if the SNP is within the open commenting region for the causal cell type, and also in the critical developmental stage, this SNP should have a large effect. So this is the preliminary result. We use the PGC data for GWAS summary statistic model training and validate in the UK Biobank. Of course, the sample size is limited. We only have 1,000 schizophrenia cases, and we random sample 1,000 controls from the rest of the cohort. This is the overall prediction accuracy. As you can see, when we don't use any annotation or just the baseline annotation or baseline plus cell type stage annotation, the prediction accuracy consistently increased. And when we stratify the PI different groups, the prediction accuracy seems mainly come from the highest risk. Although it's not the difference is not statistically significant, but it shows this trend, and we are looking to validate the results in the larger cohorts like ISAC dataset. So I talk about the three projects where we incorporate the functional annotation to improve prediction, fine mapping, and help with the implicating the cell type relevant to the trait. And I think these are not standalone aims. They affect each other. For example, if we know more about the biological context in which cell type and what developmental stage, that could better inform the incorporation of these annotation for prediction and fine mapping. And on the other way, if that indeed improve the prediction accuracy, and that's justified that identify the causal cell type and stage are truly relevant to the trait. And I think currently we are using this generalized linear model to incorporate annotation into the prediction model, but we know there must be a lot of nonlinearity between annotations. So maybe in the future, the machine learning methods could be also useful in this area. At the end, I'd like to thank my team member and my collaborators, Naomi Ray, Peter Vischer, and Ryan Lister provides the single cell data set. My colleagues in the University of Queensland, and I'm currently visiting the Aarhus University in Denmark. Also thankful for the support and friendship from the NCRR Center and the funding and the data source. Thank you very much for your attention. Sorry, I'm out of the time, and I'm happy to take any questions if I still have time.